I don't have time to go into them anyhow, because I have too many slides as usual. And I would like to talk to you about patterns and processes of diversification in cichlid fishes. The first half is sort of the pattern part, where I like to introduce some of the work we've done over the last 20 years now. I got my PhD in '88, so it's sort of like a 20-year anniversary for me to come back to the museum. And the processes part is the second half of the talk, where I'll talk about some of the work we've done on comparative developmental, evo devo type of work, and work on comparative genomics we are engaged in to try to understand the developmental and molecular basis of some of the adaptive traits that may have played a role in the diversification of cichlid fishes. So it may be heretical to say this in the MVZ, but half of the species of vertebrates are fish, which comes as a surprise to most of you, I'm sure. And as you know, they're sadly underrepresented in the museum. I think there are a few lungfish or so. I think those are the only specimens that in the museum that are fish. Well, as you know, you all are bony fish anyhow. But so this, I thought this is a little, something that I should mention, which I always felt a little as an outsider in the museum, because I worked on fish, as David said, throughout my graduate career and ever since. Cichlids are wonderful, the most interesting variety of shapes and so on. We have also worked on sticklebacks and seahorses and all kinds of other groups in the last 20 years or so, but the cichlids in particular are the groups that I have stayed with since my graduate career. And the interesting thing about the cichlids is that they are so diverse. There are probably on the order of 3,000 species, making them one of the most species-rich families of vertebrates. They come in all sizes and shapes. They have huge amount of variation in mating systems and parental care systems. In their ecology, they are extremely diverse. So they are sort of a very interesting model system to look at in terms of trying to understand patterns and processes of diversification. As a graduate student, when I was in Berkeley, I worked on a group of cichlids from Nicaragua, a few species, and I'll come back to them very briefly, although I'll focus today. I decided to leave out some of the more recent work we've done that actually dates back 20 years, but we've continued to work on these fish, and I was in Nicaragua in November last year, so we have continued to work on these fish. But I'll focus on the African cichlids. This is sort of the family relationship among the cichlids. The most basal lineages are Madagascar and India, and the neotropical and the African cichlids are respectively monophyletic, which suggests that the common ancestor might have lived something around 90 to 100 million years ago. But the pattern of diversification, African cichlids is particularly well known because most of the species, the center of biodiversity really are the East African lakes. Most of the diversity is in East African lakes, although there are several hundred species that live in the neotropics as well. In East Africa, you find species flocks, adaptive radiations in the large lakes. Lake Victoria has an estimated number of 500 to 600 species. Lake Tanganyika about 250 species, which I think is an underestimate. And Lake Malawi has 500 to 1,000 species of cichlids alone that are endemic to Lake Malawi. So you can see they really are very diverse. And of course, they also live in the rivers in Eastern Africa and in the smaller lakes. So here in the northern Rift Valley, in Lake Kivu, Edward George, Albert, you have smaller species flocks. Lake Kivu will play an important role in trying to understand the origin of Lake Victoria, and I will come back to this in a moment. So when I started working on these fish, it was not clear that we're actually dealing with monophyletic species assemblages. This is something that came out of the molecular work. Before that, people had thought that the similarities that exist between particular species in different species, flocks in different lakes, is an indication of phylogenetic relationships, of sister group relationships among fish that live in different lakes. Trying to understand the diversification of cichlids, I like to analogize them to this much less interesting group of birds, only 13 species, as you know. The key to the diversification of the adaptive radiation of the Darwin's finches has to do, at least in part, with ecological specialization and speciation, where the beaks are, of course, the main thing with which they, trophically at least, interact with their environment. So here you can see the analogy. This is from Peter Grant's and Rosalie Grant's book. And the beaks of the cichlid fishes are both the oral jaws and the pharyngeal jaws. So cichlids have modified one of the gill arches into a second set of jaws that allows them to, for example, crack snails or eat insect larvae particularly efficiently and so on. And so cichlids having this two sets of jaws to feed on different diets is presumably one of the potential explanations for the evolutionary success of cichlid fishes, because they have been able to exploit resources in their environment that other groups of fishes have not, or have been able to use them particularly efficiently. So this has been something that I've been interested in, this structure, since my PhD work, when I worked on phenotypic plasticity, trying to understand how much is the reaction, how big is the reaction norm based on the diet that these fish feed. And this is, for example, a fish that this is an X-ray that I took in the museum 20 years ago, more than 20 years ago. In the intestine, you can see cracked snail shells in this fish that has a very strong lower and upper pharyngeal jaw that allows these fish to crack snails, and only they can feed on these snails. And so I have a postdoc right now, actually, she's working with using microarrays to look at differences in gene expression between fish fed on different diets in regards to developing different sized teeth. This is not something I can talk about today, but I thought I'd mention it to sort of allude to where we've come and where we've sort of revisited old questions with new techniques. So the pharyngeal jaws is something that's important, but also the oral jaws. So here you have two individuals of a species of scale-eating cichlids from Lake Tanganyika, and they have asymmetric heads. They have heads turned to the left or to the right. And this is developmentally, Nimram is in the audience, is developmentally a very interesting thing to ask, how do they make their jaws asymmetrical? It's something we're interested in looking at. And these fish, here you have these cichlids, they swim in schools of other fish trying to sneak up on their victims. And these fish are so specialized that they will feed almost exclusively only on the left or the right side of the victims by scraping scales off the sides of the fish. So you can sort of see this here. So a fish that has a head asymmetrically turned to the right will eat the scales on the left side and the other way around. And Horry has followed this over 10 years or so and interpreted this to be a frequency-dependent polymorphism where over this period, the number of individuals stayed around 50% that are left or right-headed. And we have had these fish in the lab, and a German experiment, we put a little innocent victim fish in there, and yes, they only feed on the left or the right side. So extremely specialized. When you look at the stomachs, you find these scales in the stomach. So a lot of trophic diversity, extreme degree of specialization, and this raises all kinds of questions in terms of how did it come to this, both on the evolutionary scale and at a developmental or comparative genomics scale. To remind you of the biogeography, these are the three lakes that we've studied, and we have worked over the years using mitochondrial and nuclear markers in establishing the phylogenetic relationships. These names are not important for this talk. What's important to remember is that Lake Victoria is a monophyletic species block. That is, it can be traced back to an ancestral lineage. This is something I did in Arnold Wilson's lab as a postdoc, so we used to call this the cichlid Eve that swam into Lake Victoria and speciated into 500 species. Here represented by only 2 species. The same is true for Lake Malawi, where you have an ancestral lineage, and you have 3 or 4, I'm not showing you all of them here, lineages that live on rocks, or live over sand, or live on the open water. And then they came out of Lake Tanganyika. This lineage here, the Trophiini Haplochromini group, is from Lake Tanganyika. So the cichlids that are lake adapted and are extremely specialized, at least some of them, particularly haplochromine cichlids, were able to leave Lake Tanganyika, swim through the rivers, colonize new habitats as they became available. Lake Tanganyika is 10 million years old, Lake Victoria only 500,000, Lake Malawi maybe 2 to 3 million years old. As these new lakes arose, the haplochromines were the ones to colonize these lakes. And almost all the species of cichlids that we know belong to the haplochromine lineage. I'll come back to this later on. There are other older lineages of cichlids, but they're not nearly as species rich as the haplochromines. So there's something special even within the haplochromine cichlids, within the cichlids about the haplochromine. So this is a somewhat outdated, more pessimistic slide. We have much better knowledge about the relationships of these lineages, but the point of this diagram is to show that Lake Tanganyika is old in terms of genetic diversity, and that there are several old lineages. The one that is important are the haplochromines, out of which then Lake Malawi and Lake Victoria flock arose, and they are genetically extremely homogeneous. Again, this goes back to work that I did as a postdoc in Lake Victoria cichlid species flock. We were glad to find any mutation at all differentiating species, and on average, we'll find maybe 2 to 3 substitutions between species. But it turns out there's a large amount of mitochondrial lineage sharing between these different species. So, cichlids from Lake Malawi at Lake Tanganyika are sort of the genetic cradle, out of which then fish that colonized Lake Victoria and Lake Malawi arose and colonized these lakes. Now, as I said, before we started this molecular work, it was not clear how these species and the different lakes are related to each other. And the workers that had worked on them before had actually, on the whole, suggested that these similarities are indications of phylogenetic relatedness. But we now know that all of these fish in Lake Tanganyika are not nearly as closely related as these fish in Lake Malawi. All of these fish in Lake Malawi, again, can be traced back to an ancestral lineage, and they arose from a fish that sort of looked like this, from the Tropheus haplochromine type fish. So all these similarities between these fish from these different lakes are nothing more but similarities. So this is a very striking case of parallelism or convergence that gave rise to these very similar looking fish repeatedly in these different species flocks. And in essence, this diagram is in a way an outline of our research for the next 20 or so years, because we're trying to understand that are the same genes involved in making these fish look very similar, or did evolution invent a different way of coming up with the same phenotypic endpoint of evolution, if you will, but using different means. So we're trying to understand, are the same genes and genetic pathways involved in making these fish look similar, let's say in their dentition and coloration and body shape, or has evolution found an alternative way? So this is something that we're interested in doing, and other labs, of course, as well. So to continue with the pattern, before I get to the process part, I'd like to give you one example of work we've done more recently to try to understand the origin of the Lake Victoria species flock. Again, we're dealing with something like 500 species that live in Lake Victoria and in Lake Victoria only. They're genetically extremely similar. This is based on complete D-loop sequences. This analysis is based on something like 900 some individuals, and as you can see, we cannot tell these species apart within Lake Victoria, and they sort of blend into cichlids from Lake Kivu. Lake Kivu cichlids are shown in red, Lake Victoria species in blue. Extremely close related, except for one species in Lake from Lake Kivu. This is called Haplochromis gracilo. There it is. It doesn't look like a particularly special fish, except this is the only species in Lake Kivu that we can statistically tell apart from the superflock that includes Lake Victoria and Lake Kivu. If we graph the mitochondrial D-loop sequences in the haplotype network, in blue again are the Lake Victoria cichlids, in red are the Lake Kivu cichlids. Those are the lakes that matter. You can see that there's a central haplotype here, and there are a couple of central haplotypes that are all exclusively composed of fish that are found in Lake Kivu. Lake Kivu is a much younger lake, a much deeper lake than Lake Victoria. It's an older lake, but it only contains 18 species of cichlids, of endemic cichlids. 6 of which, I'm only showing 4 here, 6 of which have the same haplotype, and that's the central haplotype. So essentially, Lake Victoria is not monophyletic, but strictly it's a diphyletic, because this is a common haplotype found in Lake Victoria cichlids, and this lineage here is a common haplotype found in Lake Victoria cichlids. So there were essentially, as far as we can tell now, 2 lineages that colonized Lake Victoria, and they're extremely close related. They're only 2 mutations or so away from this central haplotype. We could do other analyses to try to look at the population history of the Lake Victoria and Lake Kivu cichlids. They suggest again that Lake Victoria is extremely closely related, composed of extremely close related species that are only a handful, maybe 2 or so mutations apart, very young origin of the whole species flock. We think that this species flock of Lake Victoria cichlid is on the order of only 100,000 years old or so. Based on this kind of analysis, we can reconstruct the phylogeographic history of this colonization. Again, there are two lineages that came out of Lake Kivu that colonized Lake Victoria. Later on, there were more recently lineages that were able to leave Lake Victoria to colonize, for example, Lake Kivu and so on. You can also say that the Lake Kivu cichlids, in a sort of stepping stone fashion, colonized the other rift lakes in the northern Rift Valley and started their small own species flocks in these other lakes. So based on this kind of analysis, we can get a more detailed understanding of the evolutionary history, both in time and space, that led to the origin of these adaptive variations. Just a couple of slides on our more recent work on the Nicaraguan cichlids, before I then move on to the more process-oriented part of the talk. This is a species called, right now called Amphilophus citronellus. When I was a student, it used to be called Cyclosoma citronellum. This is a species that actually George Barlow described in 1975 as a new species of a cichlid that only lives in one crater lake in Nicaragua. And these fish are very interesting both in terms of their colorational diversity. They are goldfish and they are black and white striped fish. That's why the fish is called the Midas cichlid, because of the golden coloration, based on the Greek mythological god Midas that turned everything into gold that he touched. And these fish have different pharyngeal jaws that allow them to feed on different diets. So this was actually the impetus for me to start working on DNA 20 years ago. That's why I moved to Alan Wilson's lab. Those were the pre-PCR days for those students that don't believe that people actually existed before PCR came to this planet. So I started in 1987 at Alan's lab, purifying mitochondrial DNA, doing RFLPs. Jim Campbell, remember what that is. And then PCR came and changed the world as we know it, and that's when I started working on the DNA sequences of these fish. What we ended up finding, this is some morphometric work, and this is some more population level work, using AFLP markers, microsatellites, and mitochondrial DNA. We ended up finding that the species, yes, is genetically indeed different from the ancestral lineage that gave rise to this, and so we think that this is a case of sympatric speciation. Whether or not this will turn out to be a common mechanism of how new species of cichlids arise in Nicaragua, or maybe even Africa, is an open question, but we're following up on this in much more detail with larger samples from larger number of crater lakes and so on, because it turns out that each of the crater lakes probably has a small number of endemic species that has not been recognized before, but we can tell them apart with sensitive markers and a large number of microsatellites and so on. So this is something I just wanted to mention as one of the studies that we've done to try to understand the processes of speciation. Now, the diversity in cichlids is legendary. Now we're trying to see what is the developmental and genomic basis of this. And as I said, the jaws are an important key ingredient in the evolutionary success, presumably, but what are the other factors that probably played a role in speciation in cichlids? The German word for cichlid, for the family Cichlidae, is Buntbarsch, which is a colorful perch, and typically German aptly named family. The coloration is important because presumably females, this has been shown for many species, females base their mate choice on color characteristics of the males. So coloration is something that we were particularly interested in trying to understand the genetic basis of. But of course, color is just one part of the sender-receiver situation. So visual pigments are another factor that Tom Cooper's lab, for example, is interested in, and Nori Okada's lab in Japan is interested in studying. We in other labs are interested in looking at tooth shape development and jaw shape differences. And then, of course, behavior plays an important role in all of this. So people are interested in trying to find genes responsible for some behavioral differences in relation to parental care, for example, and mating systems. This tree shows a little bit more detail the haplochromine radiation. So we have looked at this in much more detail than I showed you in the previous slide. Just very quickly to introduce the issue of color. The basic idea is that if you have variation in color, and that exists in many species of cichlids, you have polymorphisms in many species. And again, it is known that females tend to have differences for males of particular colors. So the coupling of male polymorphism and female preferences for differences in these polymorphisms might end up leading to speciation. In a nutshell, this is sort of why my lab and others are interested in looking at the coloration. Now, the bewildering diversity of coloration in cichlids can actually be reduced somewhat because there are very few, for example, cichlids that have horizontal bars. Almost all of them have vertical bars. There are certain colors that seem to be preferred over other colors. And one thing that is also very noticeable, and this is only present in the males of the Hoptochromine cichlids, they have these so-called X spots. And so one thing that we were interested in trying to understand is how are these X spots made, and because they play an important role during mate choice and all kinds of things. And I'll spend the next slide explaining to you some of the work we've done on the X spots. But how do we find genes that may be responsible for some of these phenotypic characteristics, such as X spots? One approach we and others have taken is to use candidate genes. Many of the candidate genes we've tried come from work on Medaka and zebrafish. At least the Medaka genome has been sequenced, the mutagenesis screens have been done on Medaka and zebrafish. So those screens have also been done for color pigments. And in many of the cases, the genes that are responsible for the color mutants are also known. And we can then take these color genes and look for differences between different species that differ in these colors. Another approach are QTL approaches. And the beautiful thing is that many of the species can be hybridized. Certainly within Lake Victoria and Lake Malawi, sometimes you have to use artificial insemination, but you can make fertile F2s and you can make genetic maps, as I'll show you in a moment, and use QTL approaches to identify chromosomal regions that are associated with, for example, color or jaw shape differences and so on. A number of genomic resources have been established. So we have made back libraries for several species, the Nicaraguan species, but also African species. We've done EST studies, and we've developed several microarrays based on cDNAs, for example, for cichlid fishes. And the cichlid genome project that my lab is part of is undergoing right now at the Broad Institute, where the tilapia will be sequenced and 3 species of other cichlids. So hopefully within a year or two, the entire genomes of these species will be done. Tilapia will be done at 7x and these species at 2x. The Hoptochromines, again, are this lineage of cichlids, distinct from the older lineages, and the Hoptochromines alone are something like 1,800 species. So really the success story within the successful cichlids are the Hoptochromines. 7% of all Teleos species belong to this one lineage of cichlids. And they include not only the Lake Victoria cichlids, but also the Lake Malawi species and many of the riverine species. And they can be traced back to an ancestral lineage that came out of Lake Tanganyika. One of the key innovations that characterizes the Hoptochromines, already mentioned, is the presence of X spots and maternal mouth pooling. This is something that all the Hoptochromines do. The female lays the eggs, picks them up, they get fertilized inside the female's mouth, and the female incubates the eggs inside of her mouth and takes back the young to protect them from predators. There's a color dimorphism where the males tend to have colorful colors and the females have these cryptic colors. And the X spots can be traced to the origin of the Hoptochromine cichlids. This can actually be done in a little more detail, but the X spots presumably therefore played an important role during the diversification of the Hoptochromines and may be considered to be the key innovation that is at least partly responsible for the evolutionary success of this lineage of cichlids. So what are these X spots? They consist of xanthophores, so yellow pigments. There's a mutant in the zebrafish that is known to cause a defect in these color pigments. And so this is the reason why we're interested in looking at this. This used to be called FMS, now it's called CSF1R. It's a type 3 receptor tyrosine kinase. It's a gene that may play a role in these X spots. That was at least our guess. We tried other genes also. This is the way we asked this question. We had the back library. We could identify positive clones for this gene. We sequenced several back clones entirely, and then we did in situ hybridization on probes that we developed based on these sequences. This shows you both males and females, the spawning sequence, and the presence of these X spots on the adult anal fin of the male, and the ontogenetic development of these X spots. Let me just show you how this is, these X spots are used. So it's clear, and this is a histological stain that shows that the centrophores really make up the X spot. It's not the whole thing because there's a melanophore, a black pigment ring around it. The male builds a nest and attracts the female to his nest. Females are known to base their mating decisions to some extent on these X spots. So males with larger X spots or more X spots are more sexually attractive to the females, and the size and color of these X spots mimic the color of and size of the eggs that the female is laying. So there's the female, there's the male, he's quivering. She finds him attractive, obviously, otherwise I wouldn't be showing this. So this is the female. She will lay the eggs. You can't really see this very well, but she will lay the eggs here. There they are. They're not fertilized yet. She takes them up in her mouth, and now they still need to be fertilized. Now that the role of the egg spots, you will see the role of the egg spots. The male shows her his anal fin with the egg spots. The female picks at the anal fin of the male, where the egg spots are, presumably thinking that she's picking up more eggs. The male releases the sperm, here's the vent, and the female sucks up the sperm in her mouth, and the eggs are fertilized inside the female's mouth. This is a fantastic system that ensures, for example, fertility, and makes it much harder for sneaking males to come in and so on. This is where the female incubates the eggs. Now, so we used several approaches to try to understand the role of this gene in making these egg spots. So, for example, we cut up the anal fin of males and let it regenerate. We looked at the ontogeny of the egg spots in the developing fins of males, and we looked at the presence of this gene. I don't have time to go into the history. We've worked on the evolution of this gene to some extent. Looked at the presence and absence of RNA, of the mRNA of this gene. So blue here are the in situs, the switching on of the gene in the anal fin of the male, of the males that do have these X spots. The controls, such as the females, sense probe, and so on, shows that the gene is not switched on. There, of course, the story is a little bit more complicated because the gene is also switched on in these X spots on the dorsal fin, which may be an explanation that this pathway was co-opted into the function of these X spots in the anal fin. But in any event, spots of this kind express this gene at the time when the gene is when the X spots are developing. So the in situ data show that this gene plays a role, maybe a very important role in the ontogeny of this feature. Again, all the haplochromines have these X spots. Again, the X spots evolve in the common ancestral lineage, and these are particularly species rich, so we think that maybe there's a cause-effect relationship between this mating system, the X spots, in association with female mouth brooding and so on. When we look at this gene, portions of this gene, we see that there's an accelerated rate of evolution in the ancestral lineage that leads to the haplochromine cichlid. So we looked at this in somewhat more detail, and we find that there's a pattern of increased number of amino acid substitutions in the ancestral lineage leading to the haplochromine cichlid. So the emerging pattern is consistent with the idea that there was accelerated rates of evolution that is associated with the change of function in this gene and the haplochromine lineage. The other cichlids have it as well, but only the haplochromines use it in this fashion that I showed. And when we look at the Ka-Ks ratio in this sliding window approach, again we had sequenced about 150,000 base pairs, the entire back clone that contains this gene. It's made up of 21 exons, and we looked at this gene in something like 21 species, haplochromine species, and more basal lineages of cichlids. We do find that there's this accelerated rate of evolution, and we do find a pattern where in particular portions of the gene, the NDS ratio is indicative. It's about 1, clearly about 1, as an indication that it may be under positive Darwinian selection. And these are the regions of the gene. The tyrosine kinase receptor is a receptor in the membrane of cells. This is the place where the ligand may be interacting with this tyrosine kinase. So we don't have information on the ligand, so this story is not complete, but it all makes sense in terms of the change of function in the interaction of the ligand and receptor. So to summarize this portion, so I think we have found a gene that is at least involved in an important function making these exports, and that this gene changed its function and evolved more quickly at the origin of this very successful group of cichlids. So we've also done many other kinds of studies to try to find other genes. So based on the EST studies that we've done, so we've sequenced something like 30,000 ESTs in these cichlids. We've looked at we looked for genes that show signature for positive selection. So above this line, we have genes that have the NDS ratio over 1. So there are genes that you would expect to be there, for example, MHC genes. But there's another gene that we're quite interested in called aromatase, and there's another gene that is called SDP120 that we looked in more detail. This is a gene that is expressed in sperm cells and may have to do with the function of fertilization specific to the sperm. So we've looked at this without wanting to go into detail. We've looked at this gene in the haplochromines and in other species of cichlids and find some signatures. Here are the amino acid replacements in relation to position in the gene that can be related to, in a sensible way, to the change of function. Just as one example of the work that we've done trying to find new genes. For the last 10 years or so, we've been interested in the evolution of genomes of fishes because for a while I've been interested in looking at Hox genes that play an important role during development, as you know. And about 10 years ago, it became clear that the zebrafish had more than the expected number of 4 Hox clusters. All land vertebrates have 4 Hox clusters. It turned out that zebrafish had more, and I was thought, well, maybe this is something that's specific to the zebrafish lineage because it belongs to the carp-like fishes that are known to be prone to be polyploid. But it ended up it turned out to be the case that all fishes have more than 4 Hox clusters, typically 7. Not always the same 7. And so over the last 10 years or so, we have worked on the Hox clusters of different species of fishes and ended up being able to show that there was fish-specific genome duplication that includes almost all, but not all, of the living species of ray-finned fishes. There are some basal lineages, including, for example, Polypterus, that apparently branched off the stem lineage of ray-finned fishes before the fish-specific genome duplication occurred. So at least initially, the common ancestor of all 28,000 or so species of ray-finned fishes had twice as many copies of each gene as the common ancestor that gave rise to the land vertebrates. Another reason for the fact that fishes should be represented in the MDC essays. Anyhow, so this is just a very brief overview of some of the work that has sort of developed over the last 10 years or so. We have, in collaboration with Jeff Bohr at the JGI, this goes back to 1996, started to sequence back clones of this species for which we made the back library of all the Hox clusters. And bringing up this work because one other set of questions we're interested in trying to understand is the relationship of gene duplications and changes of gene function in relation to regulatory evolution. And we think that the Hox clusters are a good model to look at this in a comparative evolutionary framework. It turns out that the phylogenetic relationships among fishes based on molecular data, such as the Hox clusters, turn out to be different than what had been thought. And of course, we have to have a correct phylogenetic framework in which we have to ask comparative evolutionary questions. So, for example, it was not thought that Medaka is so closely related to the cichlids. So this is something, based on this framework, we can now ask questions about the evolution of Hox genes. Turns out that each of the fish in which the Hox genes have been looked at, the clusters are different. none of the fish that have been sequenced so far is identical in the Hox cluster architecture. They differ sometimes in the number of cluster and the presence and absence of clusters, and based on this framework, you can reconstruct the phylogenetic relationship, the phylogenetic events that happened when particular genes evolved and were lost. So that's why you need a phylogenetic framework. The other, and again, to juxtapose this to the land animals, every frog, rat, bird, or mammal seems to have, at least for what it's known so far, identical Hox clusters. Not quite true, but almost true, and that they differ very, very little. And each of the fish is different so far. The other thing one can look at is to look at microRNAs, and as we've done here, to look at the presence and absence, changes in presence and absence of microRNAs during evolution. And as you know, the role of microRNA evolution is only recently been recognized as probably something very important in terms of phenotypic changes and the relationship of regulation that goes along with microRNAs. So quite early, so already 2003, we began to look at regulatory regions in the Hox clusters to see if there are differences within Hox clusters between different species or in different Hox clusters. One thing that jumped out at us is that among, this is based on cichlid work, the anterior Hox clusters, so 1, 2, and 3 paralogous genes in Hox clusters, tend to have a larger number, and in terms of number of base pairs, a larger amount of DNA that is used to regulate for regulation. This makes intuitive sense in the sense that the anterior genes in Hox clusters tend to do more important things, if you will, like setting up the midbrain, hindbrain boundary, whereas those things sort of more in the abdominal B region of Hox clusters do things that are not quite as important, such as appendages and penises. So here is, if things get messed up here, things can be very bad for the organism. So we looked at this in larger data sets. This is all fishes, stickleback, medaka, zebrafish, and cichlids, and so on. And you can see there's a general trend, not as clean, but as a general trend, the anterior Hox genes tend to have more presumed regulatory elements in keeping with this information. So when we look at regulatory elements and presumed regulatory elements, we can do different kinds of in silico analysis. This is the so-called VISTA plot, where you look at the Hox genes, and there's always a first and second intron and the exon, and then there are intergenic regions and so on. What you find is that the Hox clusters that are lost or duplicated and secondarily lost, you find a loss of changes in highly conserved non-coding portions of DNA that are likely to be of functional importance. So here's a peak of something like 30 or so base pairs that is almost 100% conserved between fugu, medaka, and zebrafish, for example. And in the beta copy of this Hox D cluster, you don't have this element. This is an indication that the function of this gene copy that arose through the fish-specific genome duplication has changed since this regulatory or presumed regulatory element has been lost. So this is something that we've extended to larger data sets comparatively, and as you can see, things get quite complicated. But we have compared other gene clusters, for example, the KCNA gene cluster, that's a potassium channel gene that is also maintained in evolution, but not as complete as the Hox clusters. And one suggestion for the fact that the Hox clusters have remained intact for several hundreds of millions of years has been the fact that a lot of the regulation of Hox cluster genes is both cis and trans, and so that across Hox clusters there's a lot of regulation, and that this network of interacting regulatory elements mandates that the Hox clusters do not get interrupted by other genes and stay intact. And so this is something we've compared in other gene clusters, for example, KCNA genes, where we have done similar analysis to this, where the data don't look as dense, but I don't have time to do it. Again, we've done an EST study where we've looked at a large number of ESTs, both from a brain library because we're interested in behavior, skin library because we were interested in color genes, and a mixed tissue library that included also, for example, jaw tissue. We've compared the ESTs and the cichlids to the ESTs that were known from other fish model systems to try to get an understanding, are there genes in the cichlids that evolve particularly quickly or slowly, at least have a very pronounced rate heterogeneity that may be an indication that these are genes that are worth looking at in more detail, because maybe these are genes that evolve at cichlid-specific rates. So again, the whole point of all of these painstakingly laborious and boring studies is to try to find novel candidate genes that may explain some of the evolutionary success of cichlid fishes compared to the other fish model systems that are evolutionarily much less species rich and fast. This is one approach we've taken to try to identify genes that differ in the rate of evolution, and so we end up finding some lists of genes that we can look in more detail, comparing them between different species of cichlid fishes. One other thing we've done is to construct a genetic map for this particular species that we're interested in, Vertebrate. So this is based on something like 400 microsatellites. And the thing that's important also for the cichlid genome project are the questions, for example, chromosome 3 or linkage group 3 in this species is most likely a composite of chromosome 10 and chromosome 1 in a Malawi cichlid. This is an important bit of information in terms of the assembly of the cichlid genomes that will happen, and in terms of trying to understand how easy it will be to sequence novel genomes. So we're starting a little genome center in Constance where we're getting a second generation sequencer where the idea is we can sequence whole genomes of cichlids. The genome size is about 1 gigabase. Will it be easy to assemble these sequences? We'll see. Presumably yes, because they differ very little in percent sequence divergence, and in terms of chromosomal rearrangements, we don't expect to find all that much. So there are some differences. Well, this actually is nothing, but there are some differences between the genomes of Malawi and more general cichlid. And here's the summary of which chromosomes homologous to which fish. So again, we can hybridize these fish between different species. So here we have a fish that has more horizontal stripes, horizontal stripes with a fish that has vertical stripes. We can make F1s and F2 generations, and we can look for genes that are responsible, for example, for making horizontal stripes. And this is one of the other approaches we are taking to try to then identify new genes that are responsible for these particular color patterns. Coupled with this is the search for genetic loci that tells females to like males that have horizontal versus vertical vowels. So we're also looking for female preference loci using this kind of approach. Again, the male being the sender, the female being the receiver, and a way in the system, the decider in this particular system. So the work I've presented is, of course, work of my students and postdocs. I haven't highlighted them in detail here. Paul Bungie was in my lab, he came from Berkeley. Walter Salzburger's work is something I presented. Walter is now an assistant professor in Basel. Michi Lang constructed the back library. Dirk Steinke did most of the bioinformatics work. Let's see who am I forgetting. Dave Serra did the SP120 work. Céline Clabaud did the initial work on microarrays that I didn't show you today. So thank you very much for your attention.
Other questions for Axel?
How are you examining traits that might not be under genetic control? And how do they, for instance, the stripes again you mentioned, are those really being driven by genetic control, or are they learned early in the experience of the fish? How are you going to consider those kind of
The female preference for color pattern?
yeah, preference or
that is a good question. We're not controlling for this right now. It seems to me to be sort of more, sort of a derivative question. Once we have, it will be for us, it will be the holy grail to find a female preference gene. Then we'll ask more complicated questions about social environment. It's complicated enough and enough work so far to look at this. But it's a valid question to ask because George Barlow has worked on this for decades to try to distinguish experience from genetic prevalence in terms of mate choice in these fishes, for example. So it's a very important issue, and cichlids are very smart, and I wouldn't put it past them that they will be influenced by the social environment. But right now, we're trying to keep that as stable as possible, because that's just another variable that we don't want to deal with in these kinds of studies.
Axel, Anthony Carson distinguished between different kinds of species from a geneticist, from a Drosophila geneticist's perspective. And these cichlid species, especially the white Victoria ones where you get big flocks, seem very much like what we call eco-species. That is, once you change the environment, you get cloudy water, for example, the species border sort of disappears. Just what proportion of those species are of that nature, do you think? Or are all of them essentially of that nature? They're essentially reversible species.
Yeah, that's a good and tricky question. I mean, when I, as a master's and PhD student, did the phenotypic plasticity work, I was convinced that these species, these estimates for species numbers are overestimates. I was convinced that there really are not 500 species of Lake Victoria or 1,000 species of Lake Malawi. But since some of the morphological changes that seem to be inducible by dietary differences were as large as differences between different genera, in African cichlids at least. But since I've been in Lake Malawi and Tanganyika and Victoria and dove in those lakes, I do believe much more in those species numbers, and actually think that they're probably underestimates in terms of the color diversity that is there and the kinds of behavioral differences you can see between the species. So it is a tricky situation where most of the mating decisions are done prezygotically, for sure. Yeah. And as you sort of alluded to, the visual environment at least seems to play a role in terms of making wrong decisions. If the water, if the light situation in the water changes, you might hybridize more and choose the wrong species. So I think it's still a somewhat open question, but it may be something that is more specific to Lake Victoria than, for example, Lake Tanganyika, where the species tend to be older and tend to be more phenotypically separate from each other.
So for your genomic studies, are you then taking phylogenetically more remote members of this group? Or
right, so I mean, so these decisions about which genomes are sequenced are of course very political always, and not always the best best biological reasons I use for deciding what's sequenced. But so the tilapia genome is sort of the base genome, and that's done because of the aquaculture importance of tilapia. But then the other three species that have been chosen so far are species that people have done some groundwork in terms of EST libraries and back libraries and so on. Hopefully it will be relatively simple to add more species onto this. I mean, as always, this is just the beginning, where we then hopefully, as sequencing costs go down and sequencing speed still increases, it will be possible to do more genomes. I mean, I was telling somebody today that there's a student in Svante Pauw's lab who's doing the bonobo genome for a PhD thesis. So we've arrived at a stage where a PhD student can do 3 or 4 gigabase genome as a PhD thesis. And I'm sure that as sequencing speeds and bioinformatic tools become more automated and easier to use, this will happen also for the cichlids, where presumably 1 gigabase of genome size is not going to be a deterrent for PhD thesis anymore. So this will probably happen.
How many of your students will you do?
Maybe a 4, something like that.
Other questions?
So one of the proteins that was under positive selection is aromatase. So do male and female hormones have a role in coloration?
I don't know that literature very well. Hans Hoffmann is working on this, and Russ Fernald at Stanford. They are working on this. We're collaborating with them, or have collaborated on some of our projects. For example, the EST project. The brain library, for example, was done in collaboration with them. With the intention being that this can be used for the male dominant studies that Russ are doing and Hans are doing, and hormonal changes do play a role because you can tell the mood of a cichlid, and it can change very quickly, and they can change, within seconds or at least minutes, can change the color patterns quite dramatically. So clearly, hormonal differences do play a role, and aromatase would be one of our candidate genes to look at, for example.
All right.
So you've shown that coloring patterning is obviously very involved in the evolution of the group, but I noticed that you had one slide that had an opsin gene that didn't appear to be under selection, but have you thought to look across more of these sort of eye-related?
Yeah, this is not work we are doing. Tom Coker's lab and Nori Okada's lab, they are working on the opsin genes, or I should say Carol Carlton, Tom's wife. So that's sort of their territory, and we're staying away from this. They have found duplicated opsin genes that have shifts in wavelength that are in accordance with some of the colors that are preferred in the light environment. So yes, there has been some evolution on this, but not all opsin genes, you notice this very keenly, are at least show signs of positive selection.
One of the nice things about having our former students come back and talk to us, and those of us who've been here a while can think back to the days when they were with us. And I remember very much when Axel was the TA in the evolution course. I think you and John from Bosos were the TAs the same year. So it gives me a great deal of pleasure to have you come back and now tell us all these wonderful things that are going on. Thank you very much.
Thank you.